Hello everyone, good evening and let's get started. Okay. Hi, my name is Ashwin and I'm one of your TAs for the course Drug Delivery Principles and Engineering, and this is a problem solving session, and this is week 12, so the final week of our problem solving sessions. Uh, Let me share my screen. Okay. Can someone confirm if they can see the screen?
Yes,
Yes,
sir.
sir. Okay, great. Okay, let's get started. So we'll briefly take a look at last year's week 12 questions first. Okay, so the first question reads, what is the full form of LCST?
Sir,
Sir,
option
option
B.
B. Okay, low critical solution temperature. Any other guesses? Yeah, so LCST is the lower critical solution temperature. This comes in regards to hydrogens and properties of hydrogens and where they can swell and where they can shrink, right? Okay, so with regards to that, what is low critical solution temperature? Is it the temperature at which the solution is saturated? Is it the increase in water solubility with increase in temperature? Is it the temperature at which the pH of the solution decreases? The decrease in water solubility with increase in temperature? The temperature above which hydrogens made of
Sir, option
LCST?
E.
Okay. The temperature above which hydrogens made of LCST polymers shrink. Okay. So there are multiple correct options in this. Two, I think. So what do you think is the other option?
Sir,
Sir,
another
another
option is
option is
option
option
D.
D.
Means
Means
both
both
options are
options are
correct. Means
correct.
D
Means
and
D
E
and
are
E are
correct.
correct. Okay. Yeah. So it's the temperature above which a hydrogen starts shrinking, right? Decreasing in its water solubility. And okay. So an ideal enteric coating material should have all the properties except it should be resistant to gastric fluids, it should be permeable to intestinal fluids, it should be compatible with other film coating additives, and the tablet being coated, it should have capacity to form continuous film with adequate mechanical properties, ease of application without special equipment, unstable alone and in coating solution, non-toxic with known pharmacologic activity, easy to be printed or to allow film to be applied to degauss tablets. So they're asking, give me 1 second, I think someone's mic is on. Sana, I'm going to mute your mic because there's some disturbance. That's okay, great. Okay. Yeah. So they're asking which of the following is not an ideal enteric coating materials property. Any guesses? I think someone just posted in the chat. I'm not able to see the chat, so just unmute yourself and answer or ask if you guys have it. Yeah. Yeah, so let's go through the options one by one. It should be resistant to gastric fluids. So an ideal enteric coating material should be able to do that, right? So it should be able
Sir,
to.
I
Yeah,
guess
I
it will
guess
be
it will be
option
option
A.
A. Option F. So you're saying unstable alone and in coating
So, sir,
solution.
this one,
This
the
one, the
unstable
unstable
alone and
alone
in
and in
coating
coating
solution. I
solution. I
think it will
think
be
it
the
will be the
answer.
answer. Right. Yeah. So that's the answer. So you don't want your enteric coating material to be unstable, right? So either by itself or in the coating solution, it should not be unstable. So alone, it should be very stable, and in coating solution also, it should be very stable. Only when it's entering through your gastric system, it should start degrading, uh, right? So unstable alone and in coating solution is the correct answer. Okay. Which of the following are not examples of active targeting?
Sir, D
Sir,
and
D and
E.
E. D and E. EPR effect and direct local delivery
Yes,
for.
sir. These are all EPR effect is specific targeting example mainly.
Correct, correct. Yeah, so absolutely. So EPR is the uh, enhanced permeation and retention phenomenon that's seen in cancers and most bacterial infections and so on and so forth, right? Yeah. So EPR effects and direct local delivery to the target organ are examples of passive targeting. Okay. LCST polymers are polymers which decrease their water solubility as temperature increases. This happens because at low temperatures, dash bonding between the dash segments of the polymer chain and water molecule dominates. And as the temperature increases, dash interactions strengthen. Okay. So at lower temperatures, what happens? So there is some bonding between something that dominates the hydrogels, water permeability and size, right? So what could it be? And what happens as the temperatures increase? Which interactions increase as temperature increases that's causing this hydrogel to sort of shrink?
So option C.
Option C, hydrogen. So you're saying the hydrogen bonding between hydrophilic segments of the polymer chain dominate at lower temperatures, but as the temperature increases, the hydrophobic interactions increase, and that causes the hydrogen to shrink. Yeah, so that's what happens. So in fact, if you sort of increase the hydrophobic content of this LCST polymers, right? So their LCST temperature keeps on lowering and lowering. So to a point where they can even sort of start shrinking at room temperatures, right? So yeah, option C is the correct answer. Okay. What is an example of passive targeting in drug delivery? Yeah, we've
So
looked
C
at.
and D.
Yeah, we've looked at this before. So EPR effect and direct local delivery. Okay. Which of the following polymers can be used for temperature triggered release of drugs? We
Sir,
have
A and D.
A and
Option D, sir.
D. Okay. Yeah. So PVC, polyethylene, starch, and alginate are regular polymers. They do not have a temperature-triggered release of drugs, but these two polymers, poly N isopropyl acrylamide and poly N diethyl acrylamide, they are temperature sensitive. So they have a very low LCSD temperature. So they start sort of shrinking at lower temperatures. Okay. What is an aptamer? It is a short oligomer that can fold into defined shapes, short oligomer that can neutralize any incoming bacterial DNA, molecule that acts like the immune system of bacteria, molecule that acts like
So
an
option
immune
A.
option.
Mainly it helps to form tertiary structure.
Okay, yeah. So aptamers are basically oligomer sequences that can fold into predefined shapes, right? You can create uh, like very beautiful origami like shapes with aptamers. Okay. Polyanion diethylacrylamide, also known
So
as
option A.
PDA.
Basically, it will be 21 to 32 when same things they are, so 21 to 30 will be the answer.
Yeah, so basically they're asking at what temperature, so the LCST of this polymer is it in the range of what temperature so that it's widely used as a temperature responsive hydrogel. Yeah, so 25 to 30. I guess 25 to 32 or something is the LCST range, but yeah, so that falls under uh, the range of PDA and LCST. Okay. Okay, so admin studies are useful in determining what? What are admin studies? Anyone?
Distribution, metabolism, elimination.
Pharmacokinetics.
So
Yeah.
I
Yeah.
think sir, the option C will be the answer.
Pharmacokinetics. Right. Yeah. So uh, option C. Okay, let's go through the options. Drug loading onto a nanoparticle, the efficacy of a drug against a particular disease, the rate at which drug diffuses out of a reservoir system, how a chemical is processed by the body, the immunogenic efficiency of vaccines, the aptamer with the best affinity for targeted delivery, the size distribution of nanoparticles, and none of the above. So, ADME is absorption, distribution, metabolism, and excretion, right? So, These all belong to, so these all are defining processes that happen to the chemical or the drug when you take it. So it's your body that's doing all these things, right? So absorption, distribution, specifically metabolism and excretion, these are mainly carried out by the body. So administrators basically describe how a chemical is processed by the body. Okay. Okay. So, how would you test for the toxicity of a nanoparticle? Select all the correct answers that apply. Testing the activity of nanoparticle in acellular or subcellular systems.
Sir, option A and option D.
Okay. Toxicity can be first tested in human clinical trials. Obviously, no, right? So, no. Testing the activity of nanoparticle in PBS. So, testing the activity of nanoparticle in PBS will not give you any clues as to how the nanoparticle itself is toxic to humans or animals or living systems for that matter. So, no. Testing in vitro, yeah, something that can be done, right? So you can take cell lines and test the nanoparticles effect on the depth or decay of the cell line. So that's something that you can use to test the toxicity of the nanoparticle. So option A, option D. Uh, Testing of nanoparticle activity in organ or organoid systems can be done, right? So you can have an organoid system where you can sort of test the effect of nanoparticle on the organoid itself. So organoid is basically organoid systems are basically a collection of cells to sort of mimic the function of some particular organoid. So you might have heard about this body on chip, so lung on a chip, kidney on a chip, stomach on a chip. So all these are organoid systems. So you can test the efficacy of your nanoparticle on that to see if it has any toxicity. So that's also a correct answer. Testing of nanoparticle activity in weak acids or bases will not give you much, right? So it will tell you if your nanoparticle is active at certain pHs, but it will not tell you if it's toxic. In vivo testing. So in vivo testing is basically testing on animal models, right? So mouse, guinea pigs, hamsters, monkeys. So basically testing your nanoparticle there on animal models. So that will also give you an idea of how toxic your nanoparticle is towards living systems, right? So that's also a correct option. And no testing required as nanoparticles are generally regarded as safe by FDA is not true. So all nanoparticles need to undergo testing. So options A, D, E, and G seem to be the correct answers, right? Okay. Okay. Yeah. I guess that's all I had for last year's week 12 questions. So we'll briefly take a look at this year's assignments as well. So we have a few that we haven't taken a look at. So we can try taking a look at them. Uh, let me see. People can still see my screen, right?
Yes, sir.
Yeah.
Yes, sir.
Yeah, if you guys have any questions with this week's assignment or previous week's assignments, you can feel free to ask, because I think this will be the last problem solving session we'll be having, and you guys will have a live session on 20th, which is Thursday, I believe this Thursday. So make sure you attend that. But anyways, okay, let's go through this week 11 assignment. Select the correct options with regards to vaccines for cancer. They have
So
correct
option C.
option C. By isolating cells from autologous tumor tissue, a wide range of antigens can be presented. Yeah, that's true. Uh, these vaccines are agents that can recognize and attack tumor cells. Is not true, right? That's not what vaccines are. And they have to be therapeutic, as prophylactic cancer vaccines are unsafe and cause tumors is not true. As we were previously discussing in the previous classes as well, so there are several prophylactic cancer vaccines. So for breast cancer, ovarian cancer, and so on and so forth. Okay. Adjuvants are not required for tumor vaccines is not true. Any vaccine to improve its efficacy requires adjuvants. Cancer vaccines are used for training neutrophils to attack the cancer tissue, thereby annihilating them. Partly true, but also cancer vaccines can also sort of activate macrophages, B cells, and T cells, and mostly B cells and T cells to form memory, right? So it seems to be the case that only option C is the correct answer. Okay, okay. Therapeutic cancer vaccines are a type of active dash with the aim to produce dash immune response to tumor antigens or tumor associated
It's option
antigens.
B.
Option
It's option B, sir.
B. Okay, so you're saying therapeutic vaccines are type of active immunotherapy with the aim to produce a specific immune response to tumor antigens or tumor associated antigens that are able to destroy cancer cells. Yeah, so that's correct answer. It cannot be so. Options A and D are incorrect, right? So you can't proliferate the cancer cells. So between options B and C, you would want to be as specific as possible because if you're broad, cancer cells are ultimately your cells, right? Or the individual cells. So there will be immune reaction mounted against self cells as well. Okay. Dash polymers are generally used in gene delivery. Cationic, anionic, neutral, positively
It's
charged.
option A and B.
Yeah, so cationic and positively charged particles are routinely used for gene delivery, more specifically because the nucleic acid material is negatively charged, right? So, yeah. Okay, what makes chitosan a suitable polymer as a gene carrier? Is it positively charged? Is it biocompatible? Is it synthetic of origin, hence less immunogenic, negatively charged, neutral
It's
charged, or
option A
both?
and B.
Yeah, so it's positively charged and biocompatible. Okay. Which of the following polymers are not commonly used for gene delivery? Polylysine, polyargine,
Option
PVC,
E.
chitosan, and PVC. Yeah, so PVC is not something that's commonly used for gene delivery. It's not positively charged also. So, that's that. Which of the following is false for allogenic tumor cell vaccines? Uses few established human tumor cell lines, limitless resource and cost effective, highly successful approach, risk of autoimmunity may not be effective at all. So they're asking which of the following is
It's option
false.
C.
Yeah, so it's not a highly successful approach, right? So allogenic tumor cell vaccine is basically where you sort of take the tumor cells from another individual, destroy, digest them, and sort of collect those antigens and then inject them into an next person to sort of raise immunity against those specific cancers. It's not successful at all. Okay. Which of the following is not a potential tumor immune evasion mechanism? Physical barriers imposed by tumor architecture, induction of host myeloid and lymphoid suppressor cells, blocking T cell effector function, resistance to apoptosis, downregulation of MHC, evasion of T cell recognition, upregulation of antigen processing.
It's option G.
Yeah, so upregulation of antigen processing is not a potential tumor immune evasion mechanism because if it's upregulating the processing of antigens, if anything, it will be susceptible to immune system, right? So it would in fact downregulate the antigen processing. Okay, what are not the objectives of gene therapy? Replace a missing or defective gene, repair a non-functional gene, produce therapeutic proteins that help treat a
It's option
disease.
E and D.
Yeah, so you don't want to cleave healthy genes that are already present to generate haploid cells, and you also don't want to cause dysfunction at cellular level. Okay. What property of the polymer causes proton sponge effect? Uh, is it the long chain length? Presence of
It's option
secondary?
D.
Yeah, so presence of secondary and tertiary amines is what causes this proton sponge effect inside the vacuole itself. Okay. Which of the following can be considered as an extracellular barrier for gene delivery? Inhibition of transcription by mucus, uptake of DNA by mitochondria, degradation of DNA in plasma, lysosomal degradation of DNA, endosomal escape of DNA, degradation by
Say option A and C.
option A and C.
But still, I need doubt regarding the degradation of DNA in plasma. Means who plays an important role for extracellular behavior? Is it the plasma? Means plasma. What role they play? That's having doubt.
Sorry, I did not hear you properly. Could you repeat?
Yeah, yeah, sir. Actually, they are telling the degradation of DNA in plasma, but means for plasma they act as an extracellular barrier for gene delivery. Means what's the mechanism that I couldn't understood, sir.
Yeah, so plasma contains a lot of enzymes, right? So there are a lot of DNases in plasma. So uh, the, i- so these DNases are present as a mechanism to sort of prevent any bacterial, fungal, or whatever genetic material to start uh, traveling throughout your body. So that's how DNA gets degraded in plasma because of these DNases that are present. And since it's not cellular process, it's happening in the plasma, right? So it's an extracellular process that's happening to the DNA. Yeah, so usually people circumvent this by not delivering it into the plasma, right? So you can escape this by not delivering DNA into the plasma itself. So you can sort of do localized injection to where you want the actual DNA to go, and so on and so forth. Various things you can do to sort of escape this plasma degradation of DNA. Could encapsulate them in nanoparticles and so on and so forth. But this presents an extracellular barrier for gene delivery. So if you want to deliver a particular DNA to your entire body, the easiest and most accessible approach is to basically do an IV injection. Right? But if you do an IV injection of naked DNA, it's going to get degraded in the plasma. That's what they mean. Is that clear?
Yes, sir.
Yeah. Yeah. Okay. Yeah. So that's it for week 11, I guess. So I guess we are left with week 10. Let's take a look at that also. Later we can see if you guys have any questions. Okay. Uh, in immune isolated therapy, the cell should be within dash from the nearest blood vessel to facilitate effective
Option A.
diffusion of nutrients. Yeah, so they shouldn't be too far, right? So if they are too far, there's no proper diffusion of nutrients that's happening. So ideally, the distance should be in micrometers, and the lesser the better. So usually, if cells are in the range of 10 to 50 micrometers, 500 micrometers is a reasonable distance for the cells to be to sort of facilitate this diffusion. Yeah. Which of the following would antagonize immune isolated therapy? Cells that were grafted less than 200 to 500 micrometer from the vasculature, use of biodegradable polymer to encapsulate the cell, use of mechanically stable non-erodible polymer to encapsulate the cell, use of biocompatible polymer that does not allow fibrosis on the implant to encapsulate the cells, use of polymer that can filter out antibodies and T cells to encapsulate cells, use of allogenic or autologous cells for encapsulating in the implant, use of a polymer that allows free exchange of proteins and cells to encapsulate therapeutic cells, use of a polymer that allows free passage of small proteins but not cells and larger complexes to encapsulate therapeutic cells. So what's the idea of immune isolated therapy? So there is a deficiency in the body, and you want to fulfill that deficiency, right? So you want to repair it. And so, for instance, like if a person is not producing enough insulin, let's say, so what you can do is you can sort of take the islets of Langerhans from some other patient or a healthy individual, and you can sort of encapsulate them in a barrier that prevents. So what's the problem if you directly inject these cells into the patient's body? Their immune system is going to recognize them and immediately start killing them, right? So you don't want that to happen. You want them to stay happy and produce insulin. So for that, you encapsulate them in this barrier. So what should be the property of the barrier? Should it be biodegradable? So the barrier cannot be biodegradable, right? So if it's biodegradable, the barrier is slowly eroding away or degrading away, which means the cells will ultimately get exposed to the immune cells of the patient. So you do not want your barrier to be biodegradable polymer. So this is an incorrect choice or the correct option here. And also, do you think this polymer should allow free exchange of proteins and cells to encapsulate proteins and cells? So what happens if the polymer allows a free exchange of proteins and cells? Your T cells and B cells and macrophages will immediately infiltrate the polymer, neutrophils also, and then also the antibodies that are produced by your B cells, they will also freely pass because proteins are also sort of freely exchanging. So this should also not be an ideal property for your immune isolated therapy, right? So it should be non-biodegradable and it should not allow free exchange of proteins and cells. Okay, yeah.
So option B.
The process of weakening a pathogen while generating a vaccine is called uh, vaccination, attenuation, immunization, virulence reduction, pasteurization. Yeah, attenuation. Okay. So, what are the major challenges of immune isolated therapy? Generation of immune reaction, fibrosis of implant over an extended period, fragmentation of the implant, attack by
So
CD8.
option A and
Option A and
option C.
okay. Any other cases? Okay, so yeah, so generation of immune reaction and fibrosis of implant over extended period of time are major concerns, right? So even if you're not able to generate immune reaction, well and good, but your non-biodegradable polymer is sitting there, which means it can attract a lot of proteins and cellular debris, right? So fibrosis is a major concern. Okay. Which of the following methods can be used for encapsulating cells in biomaterial? Spray drying, ionic gelation, emulsion-based methods, microfluidics, hot melt, lithography.
So option B and option A, I guess.
Sorry, could not hear you. Could you speak again? Your voice is breaking. Uh, Anyone else wants to take a guess? So the easiest option here is microfluidics, right? What is microfluidics?
Sir, mainly it involves alginate. By alginate, they form that structure like.
Correct. So need not necessarily be an alginate-based structure. Can be collagenate, whatever you want. But yeah, so basically you form an encapsulating structure and then you send your cells in along with their media and you at a particular flow, right? So the cells will naturally go and sit in the alginate or collagen or whatever and start colonizing it. So that's what microfluidics is. But yeah, microfluidics and ionic gelation are the two most uh, commonly used techniques to encapsulate cells in biomaterials. Okay. Which of the following is a type of therapeutic vaccine? MMR vaccine, cancer vaccine, polio vaccine, smallpox vaccine, HCV vaccine, rabies vaccine. Two options are correct.
So option B and F.
Okay. B, C, D, E, F. Okay. Yeah. So cancer vaccine and rabies vaccine are therapeutic vaccines. Okay. Which branch of immunity is triggered when a vaccine is administered to an individual? Humoral immunity, adaptive immunity, passive immunity, synthetic immunity, random immunity.
Option A and B.
Yeah, humoral immunity and adaptive immunity are what are triggered when you administer a vaccine, and that's how the immune response is mounted and memory is formed, right? What is an adjuvant in the context of immune response? A molecule that inhibits proliferation, a molecule that stimulates an molecule that stimulates an immune response to the target, a molecule against which immune response needs to be generated, a molecule that induces mutations, a molecule that impedes the launch of an immune response, a molecule that does not affect immune
So option
response whatsoever.
Option B, sir.
A molecule that stimulates an immune response to an antigen, right? Target antigen. Yeah. So that's the correct option. Okay. Which of the following are undesirable properties of a polymer used for immunoisolated implant? Non-erodible, mechanically stable, biocompatible, toxic, biodegradable. They're asking for an immune-isolated implant, which of the following properties are undesirable? So, like we previously discussed, it cannot be biodegradable, right? So, what could be the other property? Can it be non-erodible? Should it be non-erodible?
A and B.
Okay. Should it be non-erodible? It should be non-erodible, right? Like so if your biomaterial or the polymer that you are using is getting eroded away, the cells will ultimately get exposed to the environment, your environment and your immune system. So that's not something that you want. So it should be non-erodible. Should it be mechanically stable? So if this implant is put in the body, there'll be a lot of movement that's happening, right? So person cannot stay still. So there's a lot of movement that's happening. This movement should not cause any rupture or tear in the polymer, right? So it should also be mechanically stable. Should it be biocompatible? Yes, ultimately you're putting this implant inside the body, and the polymer has to be biocompatible, right? Otherwise, it's going to kill everything around it, which is not something that you want. Should it be toxic? No, it cannot be toxic, right? So if it is toxic, the patient is going to die. So it cannot be toxic and it cannot be biodegradable. Okay. Which of the following is a potential advantage of particle-based approaches while delivering vaccine to antigen-presenting cells? Antigen-presenting cells take up particles in large quantities and might cause the cell to burst. Co-delivery of adjuvants is possible. All kinds of antigens can be loaded. The formulation can increase the in vivo half-life of the vaccine. It allows local delivery of antigens. The formulation can be unstable. The antigen might lose integrity in the process of encapsulation. They are asking for potential disadvantages. So co-delivery of adjuvants is possible, right? But it's not a disadvantage; it's an advantage. All kinds of antigens can be loaded, yeah, to varying extents of encapsulating efficiency. But yes, all kinds of antigens can be loaded, uh, and it's not a disadvantage; it's an advantage. So the formulation can increase the in vivo half-life of the vaccine. True, right? So the main reason why we use all these formulations is sort of to increase the half-life of something inside the body, right? So that's the main advantage that these nanoparticles or particle-based approaches give us. Okay. It allows local delivery of antigens. True, right? So you can inject the particles to local systems. So if you want to inject it into the knee, you can. Not a disadvantage. The formulation can be unstable. Is that true? It's true, right? So your particle can start degrading. Your genetic material can start degrading. If you're using proteins, they can start denaturing. All those things can happen. The antigen might lose integrity in the process of encapsulation. So that's also true, right? So encapsulation is so so if you're doing active loading, uh, it's going to be very stressful for the antigen. And even if you're doing passive loading, in most cases, because there is some charge or pH or whatever that's playing the role, it might cause the antigen to lose its integrity. So that needs to be checked for, but that's one of the potential disadvantages of these particle-based approaches. Yeah. So that's all, I guess. If you guys have any questions, we'll briefly discuss. But other than that, I don't have anything else. So has NPTEL released the admit cards for the exam yet? I'm not sure. I don't think the TS know, but I'll check and we can sort of talk about it during the live session. So Rachit will also be there in the live session, so he'll be able to sort of answer these exam related questions. Any other questions?
So week 11, question number 4.
Okay, let me see. The Yeah, what makes cytosine a suitable polymer as a gene carrier? That's the question.
Yes, sir.
Yeah. What what's your doubt? So the options read positively charged, biocompatible, synthetic origin, hence less immunogenic, negatively charged, neutral charged, or non-biodegradable. So chitosan, the main reason it's suitable as a as a gene carrier is because it's positively charged, and also it's biocompatible because it's not something that's synthetic, right? So it's a naturally occurring polymer, and it's biocompatible. It does not cause toxicity to mammalian systems.
Okay, sir. Thank you, sir.
Yeah. Okay. If you guys don't have any other questions, we can end the session here. Good luck with your
Sir,
exam. Yeah.
excuse me, sir.
Yeah.
Sir, is there same question MCQ pattern for final exam also? That means more than four options for any question.
Yeah, so ideally there should be more than four correct options per questions. So yeah.
Sir, one doubt. Sir, regarding what's the essay based question means in website it was showing.
Sorry.
It was written that MCQ online based exam, then it was written in bracket essay based. Essay based question means what sort of they will ask.
I don't quite MCQ means what else is there?
MCQ fill in the blanks and essay based.
Essay based questions. Okay.
Yes.
So so essay-based questions most likely they can ask you to define something or so mostly for our course at least most probably you'll get numerical questions and you'll be asked to show the steps, but you can also be asked to define something or maximum one or two sentence answers. That's it. There's not much.
Means for essay based question, we have to write the answer or options will be given for the definition part.
So, so I'm not sure if there are essay type questions for this course. If there are, you will need to write and scan and upload.
Okay, sir. Means actually it was written in bracket, so that's it.
Yeah, yeah. So at least for this course, the essay type questions are one or two sentence answers, and if that's the case, you need to write and upload. But let me check back and get back to you on that. Did people not tell you the final exam format?
No, sir.
Gururaj or something? Okay. Okay. Let me check back with Rachit and we'll tell you guys like what the final exam format is. Hopefully on Thursday. Your final exam is on 28th, you said, right?
No, sir. 30.
30. Okay. Yeah. Okay. Let me note this down so that I can bring it up. Okay. Anything else? Okay.
Sir, After this meeting means today is the last problem solving session. Sir, will there be actually means meeting regarding any exam discussion regarding
Yeah, there is a live session on 20th. So,
means same time only 7 to 8.
sorry?
Same time only, sir, from 7 to 8.
No. So the live session is, I think, 5 to 6. Let me check. 4 to 5 or 5 to 6.
4 to 5, sir.
Yeah, 4 to 5, I guess. Yeah.
Okay, sir.
On 20th
20th of
October, sir.
October. So day after tomorrow, there'll be a live session 4 to 5. Rachit will be there. I'll also be there. So you guys can please make sure you join, and we can discuss things. Anything else? Okay. If that's the case, then okay. Good luck, guys, for your exam, and it was a pleasure having these sessions with you guys over the past 12 weeks. So hopefully you all will clear your exam with flying marks. All the best. Bye.
Thank you, sir.
Thank you, sir.
Thank you so much, sir.
Thank you, sir.